So this series of lectures is going to look at single factor designs, and we're also going to look at how we analyze a data set to test a hypothesis. So before we begin, let's just make sure that we have a good understanding of what experimental design is versus what data analysis is. So the experimental design is really the process of planning out a project or a study to meet specific objectives and to test specific hypotheses. And so everything that we've been talking about up to this point, the way that we address principles of experimental design, the way we install experiments, the way we look at descriptive data, is all a part of that planning process. And so we know that if proper planning occurs, that the right type of data and a sufficient sample size and power are available to answer the research question of interest. And we can do that as clearly and efficiently as possible. The flip side of this is the data analysis side of it. And so experimental design and data analysis go hand in hand, but they are not the same thing. Data analysis is the process of taking raw data or transforming raw data collected on a study into and turning it into usable information. And so again, experimental design will inform the data analysis, and the way that you analyze your data will inform the experimental design that you're going to use. So let's look at an overview of single factor designs. There are three main ones that we are going to describe fully in detail. And the first one is the simplest of them all. It's a completely randomized design. And a completely randomized design can be analyzed using a couple of different tools. The one that we are going to use the most is going to be a one-way analysis of variance. And so the analysis of variance, are abbreviated as ANOVA. You may also hear it called single factor ANOVA, is going to be the primary way that a data set will get analyzed. And so other single factor designs would be a randomized block design, which adds one more layer of known information into the design. And then the last one is going to be a Latin square design. And we're going to talk about all three of those. So let's start with the simplest design, which is completely randomized. And so when we go to determine or analyze a data set that has been set up to use a completely randomized design, T becomes the treatment. So T is going to signify the treatment means. And so one of our first steps is to solve for T with the data set. So with a completely randomized design, the treatments are randomly allocated to experimental units. So that can happen by drawing out of a hat, by doing a gate cut if we are dealing with animals, by using ping pong balls, or by using the randomization tool in Excel. And so with a completely randomized design, it is our goal to make inferences about corresponding treatment means. So some of the advantages to this CRD is that they are very easily usable. And so it's easy to construct, easy to pull together, the data is easy to analyze, and it can be used for any number of treatments. Some disadvantages are that the completely randomized design is really best suited for research situations where there are a few treatments. So, like probably 1 to 5 treatments is going to be the best size for a completely randomized design. Another disadvantage is that the experimental units to which the treatments are applied to has to be as homogeneous as possible. So that means that in the context of like the cattle mineral study, where grazing location is our treatment, but our experimental unit is the cows, the individual cow, that those individual cows at each grazing location must be as close to the same as possible. And so they have to be of the same breed base, the same physiological age, the same maturity, and eating the same grasses. And so we try to make those experimental units the same to get rid of any exogenous or extraneous sources of variability that can inflate the error term that is going to be calculated as part of this analysis. So if that error term is inflated, it makes it a lot more difficult to detect the differences that are actually associated with the treatment and not with those outside factors. So with a completely randomized design, in order to analyze the data, we can do that one of 2 major ways. And there's more ways to do it, but for the purposes of this class and your first introduction to experimental design and data analysis, we're going to analyze the data 2 major ways. We can either use a t-test, which is going to use a t-statistic that checks if 2 means, so a mean from treatment group 1 and a mean from treatment group 2, are reliably different from one another. The other way we can do this is using a one-way analysis of variance, also called a single factor ANOVA. So both of these analyses with a completely randomized design data set will generate the exact same results as long as the design is a single level or a single factor experiment. And what that means is that we're only testing one set of treatments. So for example, in the class projects, the cattle mineral study and the range fire study are both single factor treatments, where we're only looking at the grazing location in the cattle mineral study, and we're only looking at burn status in the range fire study. So there's some good resources for how to conduct both of these data analyses on YouTube, and so I've listed those here. I'm going to teach you guys how to do a one-way analysis of variance. We're going to briefly go over how you calculate it by hand, and then I'm going to actually show you. You guys are going to get to practice using the computer modeling system in Excel and a couple of online tools to do a one-way analysis of variance. Alright, so with a completely randomized design, what we are trying to generate is a couple of statistics. And so, first of all, we are looking to be able to partition the total sums of squares. And so the sums of squares is something that you calculate as part of the variance descriptive statistics. So if you think back to the descriptive statistics in the intro to statistics lectures, we hand calculated variance for a problem. And so those principles are actually going to be used in this data analysis process. And so with a CRD, one of the statistics that we are going to calculate is total sums of squares, or SST. And so this, sums of squares of the treatment, or SST. And so SST is where we will actually add up all of these squared values between the sample treatments. And this will give us a measure of the variability of the sample means compared to the overall mean. The second statistic that we're going to calculate is the sums of squares of the error. And so this is where we add up all of the squared values within an individual sample. So relative to the class projects, this would be the sums of adding up all of the squared values within the burned plot treatments, or within the unburned plot treatments, or within the Shasta County grazing location on the cattle mineral study, or the North Chico or the South Buttes treatment. And so those sums of squares within the sample are what we refer to as the sums of squares of the error. And this value measures the within sample variability about the sample mean. So sums of squares of the treatment measures variability between samples. Sums of squares of the error measures variability within a sample. So all of these experimental designs are equated to a mathematical equation and a statistical equation. And so this is the simplest and the most basic equation that we use for a completely randomized design. When we move to a randomized block design or a Latin square design, the equation gets longer and more complicated. And so this model, the statistical model, is set up very similarly to the traditional mathematical equation for slope. So y equals mx plus b, where y over here is going to be the mean of the dependent variable or the response variable that we are interested in. Okay, mu is going to be the overall mean. This signal here, I think this is an ellipsis, I can't remember, is going to be the effect due to the treatment. And then E over here, or sigma, is going to be the random error associated with the treatment group. And so this is really going to set up the basic model, and it is actually going to be directly correlate to how the answers are presented, or how we figure out whether we have significance between two treatments or not.